Alright, so let's review gene linkage. Okay, before we start with that, let's take a look at Thomas Morgan's experiment with fruit flies. Okay, so this goes along with the PowerPoint. I just kind of wanted to draw it out or write it out. He took a look at two different types of traits between those fruit flies. One was taking a look at body color, so we have gray body was the dominant trait versus black body, which was the recessive trait. He then also took a look at, normal size wings, which was the dominant trait, versus like shorter vestigial wings, which was recessive. Alright, so what he did was he took a look at two parents. One was going to be a heterozygous for both traits. So heterozygous for gray body, also for normal wings. And he actually crossed it with homozygous recessive for both of the traits. And he wanted to see what was going on with those traits and how they're going to be passed on. Now, instead of doing like a 16 square Punnett square, we can actually make a much smaller one. So in this case, if we just take, let's say, this 4 boxes, one parent is only going to have, there's only one way of passing down the G allele. So they can only, whenever it makes its sex cells, they can only pass on the lowercase allele. Here, they can only pass on the lowercase allele for also wings. For this parent, when they make sex cells, they can have different combinations of them. Now remember, you could use foil or a smaller Punnett square to kind of see what are the different combinations of alleles that would be in their sex cells. So here we can have both of them capital. We can have both of them lowercase. Or we can go ahead and have one that's lowercase and with a capital allele, and then the opposite. Now, when we went ahead and did a Punnett, filled out this kind of modified Punnett square, it would have these different offspring. And when it had these different offspring, they could have, each one had an equal percentage of turning out to be the offspring. So out of the 4 boxes, this had a 1 out of 4 chance, or 25%, chance of turning out to be heterozygous for both traits. These two parents can have a 1 out of 4 chance, or 25%, of having homozygous recessive for both. And then here we have both, again, 1 out of 4, 25% chance of having kind of a combination of the other. Now here's the thing, these two right here, when these gametes were used to go ahead and create the offspring, it created the offspring's genotype that looked exactly like one of the parents. So here's heterozygous for both. That looks exactly like one of the parents' phenotype. It basically is the same thing. Here is the other phenotype, or genotype in this case, that the offspring can have, and it matches the same thing as what the parent is. So the phenotype for gray body and vestigial wings, or black body and vestigial wings, and gray body and normal wings, they have the same phenotypes as the parents. We call these, parental phenotypes, because they match the same phenotypes as whatever the parents are. These two, since they're not exactly like the parents, they're kind of a combination. They've been recombined, to form these two genotypes. And because of that, they don't have the same phenotypes as one of the parents. We call them recombinant phenotypes. Okay, so the expected outcome was in this kind of modified Punnett square was going to be each one was supposed to have an equal chance of becoming of a 25% chance of having one of 4 of these offspring. Okay, but what happened was when Morgan went ahead and actually saw the fruit flies, okay, and saw the phenotypes of the offspring from these 2 parents, he saw that the percentages were a little bit off. Instead, he saw 42% of the offspring had this genotype and the phenotype of gray body and normal wings. Okay, and about 41% of the offspring of these 2 parents had this genotype with black body and vestigial wings. And for the other, here we have this genotype. There was about 9% chance of all offspring turning out to be like that. And in here there was an 8% chance of all of the offspring turning out to be like that. So this is what he actually observed, and it didn't match. So he was trying to figure out why didn't each one of these phenotypes have the same or equal opportunity to be shown in the offspring when they all have a 1 out of 4 chance of doing that. So we have to kind of take a look at these genes, whether they're linked or unlinked. Okay, when genes are unlinked, that means these genes are located on different chromosomes. Okay, and because they're unlinked, we're going to show why, we can kind of prove here, well, if they're on different links, they can all have an equal opportunity of showing up onto the offspring. So let's take a look. Let's pretend this is a cell that's in an organism. Remember, half of our chromosomes come from mom, half come from dad. Okay, so mom will give like a big chromosome and a little one. Dad will give a big chromosome and a little one. Okay, let's say for a specific trait, okay, mom can give an A trait, okay, and a totally different trait that's on a totally different chromosome. Mom can also give this B allele for this trait, okay. And then let's go ahead and say that dad, okay, these are homologous chromosomes. They're the same size, same shape, so they're going to hold the same information. Instead of having a dominant allele for that trait, maybe he's going to give a recessive. And instead of giving a dominant allele for this different trait on this different chromosome, he's going to give the recessive. Okay? And when interphase occurs, okay, remember that we're going to have these chromosomes. They're going to be duplicated or copied. So here, what we're going to have is mom's chromosome is going to go ahead and just be copied again. Okay, and her smaller chromosome that has the B trait, whatever it is, she gave a dominant allele, so it's going to also have, it's going to be copied and have a dominant allele too on that copied chromosome. Dad's information is going to be also copied, so he gave the A, lowercase, a recessive allele, so it's going to be copied. And on the different chromosome for a different trait, okay, he's going to give the recessive allele. All right. Well, when meiosis occurs, now then they're going to go ahead and do prophase 1 all the way to prophase 1, metaphase 1, and so on until we get 4 cells or gametes. Okay. There's going to be different combinations of these different cells. Okay, so when meiosis happens, okay, let's say mom will go ahead and give, okay, the A allele and also maybe that B allele too, and they're on different chromosomes. Okay, let's say one of these A's, okay, will go ahead and get separated into this next gamete. But let's say maybe a B, a recessive allele B is from dad. Okay. All right. So then we have maybe here we have these chromosomes are going to go ahead and be put into one cell. So a recessive A and a recessive B. Okay, and then maybe we have like a different combination of them where we have maybe the recessive A is going to be there, but the dominant B is going to be given by the mom. So because of this, okay, it'll show that there's 4 of these cells, okay, they're going to go ahead and be made, and you see that the chromosomes are kind of all split up into different gametes, and each one will have a 25% chance of being formed with these kind of combinations of the chromosomes. Okay, so remember you get one set of chromosomes from mom, one from dad. Okay, they duplicate and then they kind of get distributed into these cells. Okay, now all of these gametes are going to have an equal possibility of having these combinations. Okay, and this is what the law of independent assortment pretty much said. Now the thing was, okay, that's what we were kind of trying to say, okay, that each one of these combinations has a 25% chance or 1 in 4 chance of being shown in the offspring, but that's not what really happened. So what are linked genes? Linked genes are located, okay, on the same chromosome. So instead we have, let's say, okay, mom will go ahead and give an A, and instead of the B being on a different chromosome, it's on the same chromosome. Okay, dad will give his chromosome, and he'll give the recessive, but that B, instead of being on a second chromosome, it's on the same chromosome. Okay, when interphase occurs, and we have a copying of the information. Okay, so let's say here we have mom's information gets copied, so we have a duplicate of it. Dad's information gets copied, and you see, I'm sorry, that should be a lowercase a, and you see that both of the traits or genes are on the same chromosomes. Okay, and now the thing about it is, when crossing over occurs between homologous chromosomes, then what we're going to go ahead and see is, all right, well, maybe one of this that wasn't crossed over, okay, this chromosome will go on to one of the gametes, so A and B, okay. Let's say this chromosome will go ahead and go to one of the gametes. And then these middle ones, remember, they crossed over, they kind of swapped pieces from each other. Okay, so we have maybe a dominant, and the dominant A will go ahead with the recessive B. Okay, and the recessive A will go ahead and link up with the dominant B. And what happened here was these were due to crossing over, right? The other ones, the first two, not crossing over. They just stayed the same. The traits are on that same, or genes are on that same chromosome. And what we saw was that these two, okay, were seen more often. Okay, more than 25% of the time they were being seen in the offspring. Okay, and these two combinations, okay, in the cells, they weren't seen as much. They were actually seen less than 25%. Okay, so the gametes that were happening, that were being made with these different combinations of alleles, were actually happening, were being created less often. Okay, more of these sex cells, sperm or egg, were having these combinations, okay, that didn't have to deal with crossing over, and the ones that were crossing over, these chromosomes, okay, were not being seen as much. So, we can kind of see that over here, where the parental genotypes were kind of seen more often, okay, than the one that were being recombined. We don't see that very often. And that was proof that the genes, okay, were on the same chromosomes. Instead of having it be, the genes be on separate chromosomes, okay, because if they were on separate chromosomes, each gamete or sex cell would have an equal opportunity of having any of these combinations, having these chromosomes with these combinations. But the fact that these two, instead A and B, they're not on separate chromosomes, they're on the same chromosome, and because of crossing over, now there's kind of like a difference in how the gametes are kind of spreading out the information. Okay, you will see instead of having it 25% of the time being all equally spread out, you see more often these, the ones that were not crossed over, we're seeing more often in gametes than the ones that were crossed over. So that shows that chromosomes have genes that are on the, well, genes are on the same chromosome sometimes. Okay.